afternoon, everybody. This is the third talk that we have of this STEM week. I'm very pleased to present Dr. Elvira de Mejia. Dr. de Mejia obtained a PhD in plant biotechnology and master in food science and technology, and she's a biochemical engineer. She's currently a professor in the Department of Food Science and Nutrition, Human Nutrition, as well as the Director, Division of Nutritional Science at the University of Illinois at Urbana-Champaign. Her most significant projects involve extrusion process, allergen reduction, molecular science, and immunoreactivity of ethanol extracted soybean protein, among other food research-related projects. So, Dr. de Mejia, can you hear us?
Yes, I can hear you, Miguel.
Yes.
Thank you very much.
Okay, thank you.
Thank you very much for the introduction. Good afternoon, all, and thank you to Dr. Federica Catti, Associate Professor and Chair of the STEM Week Committee, for the kind invitation to participate here with you at the Arkansas State University Campus Querétaro. I bring you greetings from the University of Illinois.
Thanks to you for accepting our invitation. We're really excited to hear about that.
Thanks, Dr. de Mejia, for accepting the invitation.
Very welcome, very welcome. Thank you. Okay, before we start, I just want to acknowledge two students that were the leaders of most of the presentation that I'm going to share with you today. Now Master in Science, Regina Cortez, a food scientist, a food chemist, and Master in Science, Rebecca Kowalski. She's an associate scientist here at a company in Wisconsin. So thanks to both of them for all the work they did during their master's degree and for all the publications and innovation and knowledge that we were able to obtain with their efforts. So let's get started talking about the effect of fermentation on phenolic composition and antioxidant capacity of blackcurrant varieties and beverage. In the next slide, I present the outline of the talk. Let me see if it's working. Here is the outline, among the different plant foods that provide bioactive compounds in a healthy diet, there are the berries. So I'm going to talk about blackcurrants today, their health benefits, commercial products, and some of the most important bioactive compounds in these berries, which are phenolic compounds and anthocyanins. And then I will bring you to alcoholic fermentation and what chemical and metabolic transformation took place during this alcoholic fermentation, as well as later during a lactic acid fermentation, and some summary and recommendations. Introduction. So blackcurrants are emerging as a trendy new flavor, an ingredient with many potential health benefits. The goal of this research has been to understand how blackcurrants might be utilized to provide some solutions to meet the consumer's demands. We believe that it is possible to meet these goals, particularly in the case of natural pigments. What you see here is where here at the University of Illinois in Urbana-Champaign, we have the greenhouses and we have all the farms and we can collect the blackcurrants. Health benefits of blackcurrant in humans. Rebecca Kowalski prepared a very interesting review published in Antioxidants in 2020, where she shows that there are scientific evidence regarding neurological conditions, the benefits of blackcurrants on neurological conditions such as Parkinson's disease, benefits on cognitive benefits and reduce anxiety and greater alertness, and also improve performance. So I invite you to take a look at this publication in Antioxidants, where you will be able to witness some of the scientific basis of health benefits of blackcurrants. current products, you can see that there are different products, and just recently the journal Flavor Insight reported that there are more than 3,000 new products appearing globally. Blackcurrant is a flavor to keep an eye on, and while blackcurrant's commercial products, especially beverage, are quite common in Europe and Asia, not in the United States. So there are opportunities to the food industry because blackcurrants tend to be much more astringent than other berries such as blackberries or blueberries. And just in 2016, the company Pepsi released blackcurrant cola. So there are different opportunities to understand better what are the qualities of these berries. Regarding the chemical composition, here I present some values, but please remember that these are approximate percentages because it depends on the variety of the berry, the environment where the berry was grown, but also on the degree of maturity, the ripeness. But in general, we're going to say that they contain about 80% of water, 15% of carbohydrates, 1.4% of proteins, and a certain percent, less than 1% of lipids. And you can see here also the sugar content and the main sugars that are present, as well as the acid content, the organic acids that are present, citric acid, malic, and ascorbic acid is one of the main components of these berries that you will see later in comparison to other berries. Because we are going to be talking about phenolic compounds and flavonoids and anthocyanins, I want to put it into perspective, the plant food bioactive compounds. There are many food plant-based bioactive compounds, and here is the big family of polyphenols, and under those are the flavonoids, and we will be talking today about flavonoids as well as anthocyanins and anthocyanidins that are present in different foods, but including berries. So you can see here different phenolic compounds, but especially the anthocyanins that we will be talking about. So blackcurrants contain very powerful bioactive compounds. Anthocyanins, tannins, and other phenolic compounds potentially reduce the incidence of communicable, non-communicable diseases such as type 2 diabetes. As you can see here, anthocyanins belong to the flavonoid class of phenolic compounds. The 6 main anthocyanins found in foods are mentioned here, and the structure of these anthocyanins can affect their health benefits. What is their health benefit? Well, it depends on the structure. For instance, delphinidine linked to a sugar called rutinoside. So delphinidine 3-or-rutinoside is one of the main anthocyanins present in blackcurrant that has been reported to have health benefits. How do blackcurrants compare with other berries? Well, we could say they are more astringent. They are very common in Europe and in Asia, and very high in anthocyanins. As we can see here, when we compare blackcurrant, blueberry, and blackberry, we can see that the concentration of anthocyanins in milligrams per 100 grams of dry weight are significantly higher than the other 2 berries, and also high in polyphenols. So high anthocyanin or proanthocyanidin concentration than blackberries and blueberries. Another fact about composition is that blackcurrant contains higher concentration of ascorbic acid than raspberry and blueberry. And here I want you to see different minerals and what is the composition in comparison to other berries. So high in calcium, iron, potassium. In general, these are higher berries than other berries that we usually consume. So interesting compounds in these berries. We're going to talk about fermentation today. And what is fermentation? Well, here I thought to share with you fermentation, for instance, the metabolism of glucose and what happens when we have certain microorganisms that will promote the fermentation, for instance, lactic acid fermentation or alcoholic fermentation. So today we're going to be talking about both the fermentation of blackcurrants with using lactic acid bacteria or the alcoholic fermentation using yeast. So here are some examples of the products after the fermentation or the alcoholic fermentation here to your right. So we are going to be talking about these two today, and what are the benefits, if any, of these processes on the bioactive compounds that are present in our foods. Some of those important compounds have antioxidant capacity. What does that mean? That means that these compounds can reduce or can scavenge free radicals and reduce oxidative stress. And that's very important in our organism, in our cells, to prevent from oxidation of proteins, genetic material. So having a diet rich in antioxidants is important. And here, in some researcher, this was not done in my laboratory, but it's important to look at what happens when we ferment blueberries and blackberry with lactic acid bacteria. So what these researchers report is that the antioxidant capacity increased due to the presence of certain enzymes in the microorganisms that they use to ferment blackberries in this case. Here you can see the increase before fermentation and after fermentation of the concentration of the antioxidant capacity when they use certain bacteria, in this case Lactobacillus plantarum, the antioxidant capacity increase. And remember again, is a positive fact that the biotransformation of organic acid to lactic acid could give us higher antioxidant capacity. This will increase the health benefits of the product. Also, in my laboratory, now Dr. Michelle Johnson, she's a scientist in one big company, tested the combination of blueberry and blackberry in vitro and in vivo. And what she found was that after fermentation of these two berries, the oxidative stress was reduced in animals, inflammation, markers of inflammation were decreased, and she also demonstrated the anti-diabetic effect of some phenolic compounds present in fermented berries, and what was the role of flavonoids to improve human health. So it is very important to associate this improves with, in the case of Dr. Johnson, the increase of insulin secretion and the decrease in glucose levels. So as a summary of her work, she found that the fermentation increased markers of inflammation, anti-inflammatory properties, reduced oxidative stress, and stimulated insulin secretion and decreased glucose absorption. So remember, consumers are increasing the demand for healthful foods that are not just calories, right? But also provide maximum nutritional benefits. There is also the additional demand that any additives in processed foods have a dual functionality. It's not just the physical functionality of good color or taste, but also serve as a means of adding value, adding health benefits to the products. So the goals of our project were to fill the gap in knowledge regarding the methods of extraction of blackcurrants, explore green food safety methods using water, and understand how enzymes and the fermentation process affect concentrations of bioactive compounds such as phenolic compounds, and what was the effect of antioxidant capacity. So let's start with alcoholic fermentation. The specific objective here was to characterize blackcurrant berries to evaluate their bioactive compounds, specifically phenolic compounds, and what happened after the extraction, the water extraction using ultrasound-assisted extraction. What about if we use enzymes to better liberate bioactive compounds before the fermentation of the products. So that was the objectives. These were the objectives of this part of the study. The materials came from the University of Minnesota and some others from Illinois. So we compared 4 varieties, and the extraction were carefully performed. Of course, first the characterization of all the parts in the berries, what happens to the seeds, what is in the seeds, what is in the whole berry, what is in the skin, and of course the juice, what is in the juice, and what characteristics we can separate in the different components of these important berries. All the samples were freeze-dry, and we use a mortar to grind the samples before analysis. Like the big picture experimental design we use here. So 4 varieties, of course, each of the juices were collected, frozen, and freeze-dried. And then the remaining solids were extracted using this ultrasound-assisted water extraction. And before fermentation, we use some pectinases in order to liberate more bioactive compounds, such as antioxidant compounds, such as phenolics, anthocyanins, proanthocyanidins, liberate them from a sugar matrix of the pectin matrix, and then the fermentation happened. Today, I'm going to share with you results on anthocyanins, proanthocyanidins, polyphenols, antioxidant capacity. I'm not going to talk about the biological activities in this presentation. So yes, we use an ultrasound-assisted extraction, mainly with water, and this produces an acoustic cavitation effect on the solvent, and that makes that the molecules move and the release of compounds is accelerated. So these are some of the molecules that we were looking for, and the extraction was accelerated using this ultrasonic or ultrasound-assisted extraction with water. So we did it for 2 hours. Then the remaining solid was again extracted for 2 hours, and again the remaining was extracted for another 2 hours in order to make sure that we didn't have any important components left over in the matrix of the fruit. Then in preparation for the fermentation, we use pectinases. Different juices were processed with pectinases to maximize the release of anthocyanins and proanthocyanidins from the matrix. As you can see here, the role of the pectinase was to separate, split this cleavage of the glycosidic bond in order to split phenolic compounds and liberate them to make them available for fermentation later. And here I want to stop and comment on the importance of collaboration with industry. We collaborated with this industry where they provide funding for the research, but also in-kind provided special enzymes that they were developed just recently, very, very new materials that we were able to test. So interacting academia and industry brings synergistic results, and it's very important to do it. So just to show you the pectinases that they provided, and we were very grateful because these were newly developed by the company, and we tested them before we actually fermented the product. So you can see here the small experiments where we started doing in a small scale, testing different concentrations of the pectinases, different times of exposure, and see which ones were the most adequate for our fermentation. And then the fermentation of black currants to make wine. And because this is important for people with type 2 diabetes, we at the end de-alcoholize the wine, but all the bioactive compounds remain in the wine prepared. The yeast were causing the release of more phenolic compounds and transformation, metabolic transformation of all of these phenolic compounds, and we were able to chemically and biochemically characterize the products after the pectinase treatment, and of course after the wines. So here is just the different steps that we took in the process of the fermentation. This is alcoholic fermentation again. So here you can see the details of the different varieties tested. We did a mixture of some of these three varieties of blackcurrants and compared with one of our best varieties in Illinois. So this came from Minnesota, and this was one of our very best variety from Illinois. We use different amounts and times of exposure with the enzymes, and then we use this star yeast again, collaborating with industry to obtain this very best Saccharomyces cerevisiae for the fermentation. This was very fast starting strain that produced round and smooth, more aromatic wines that mature very quickly. So we were able to confer with the companies and obtain this very good star yeast for our fermentation. I have all the details if one would like to see the details of our fermentation. And then dealcoholize, remove alcohol, and kept all the bioactive compounds in these preparations. And then characterize all the phenolics present, as well as what was the effect of the different steps during this processing. First of all, let me share with you the results of total polyphenols and condensed tannins. These are milligrams equivalent of gallic acid per gram of dry weight of the material for the 4 different varieties tested. And as you can see, blue is the whole berry. Then we have the orange, the juice, green seeds, and orange, strong orange here, the skins. It's important to indicate that seeds and sometimes also skin contain high concentrations of phenolic compounds. To the right of the slide, we can see the condensed tannins in milligrams equivalent of catechin per gram of sample. And here we can see that all the varieties have very high concentration of condensed tannins in the skins, as well as, of course, in the whole berry, but the skins still have interesting material that could be isolated and utilized. Okay, so now we're going to talk about the different steps during the pre-fermentation and after fermentation. Here you can see the total anthocyanin concentration in milligrams equivalent of cyanidine 3-glucoside per gram of sample. And the first one, the red one, is with no heat or no enzyme. And then as we move to the right, we have without pectinase, the first group, the second with pectinase, 400 milliliters per ton, and etc. So we have different concentrations. So in this case, you can see that we can optimize the amount of enzyme added before fermentation to improve the liberation of bioactive compounds. So this can be optimized and obtain which one would be more convenient for whatever objective we have. And to the right, you see post-fermentation. You can see here that the mixture of different berries gave us higher total anthocyanin concentration. And it was also interesting to see that the leftover material after fermentation still had anthocyanins. It's called the pomace, right? The solid material that is left over after fermentation. So we have coproducts here that now can be used for other purposes, but they still contain a high amount of anthocyanins. What about total polyphenols? Well, it was surprising to see that total polyphenols increased significantly post-fermentation. Either the wine was dealcoholized after fermenting at 23 room temperature or at 15 degrees. 15 degrees seemed to be better than 23 degrees temperature, but the concentration in milligrams equivalent to gallic acid per gram were very significantly higher than all the rest. Again, the pomace, the solid material left over after fermentation, also contains total polyphenols that may be interesting to use this coproduct after fermentation. So what we are learning here is that we are finding different compounds after fermentation, And we use liquid chromatography to determine which compounds were present, and we did identify delphinidine triiodrutinoside. You can see here the structure and how it looks at 520 nanometers and at 280 that detects phenolics in general. So you can see the profile of these chromatograms and the presence of these compounds. Of course, we had some pure anthocyanins, and that's why we could determine, but you can also use mass spectrometry to identify the nature of these compounds. And here you can see the different compounds, delphinidine triiodrutinoside, one of the anthocyanins present in wine and in berries, and also cyanidine triiodrutinoside. Okay, so finally, we're going to see that in all the preparations, we had very high concentrations of delphinidine triiodrutinoside in the pre-fermentation and post-fermentation. This was one of the main anthocyanin present, which is very good because that's one of the most bioactive anthocyanin. And in the next slide, we can see some correlations. So we need to optimize the process before we produce any product. So in this case, we notice that the total condensed tannins and total polyphenols correlated with the time of exposure of the pectinases at 52 degrees centigrade. So as we increase the time, more total condensed tannins and total polyphenols were extracted. And regarding the fermentation, we also saw subcorrelations for total anthocyanins, condensed tannins, and polyphenols regarding the temperature of fermentation. So these are parameters that need to be optimized every time that you use different products and try to get the most out of these enzymatic and fermentation processes. So in conclusion of this alcoholic fermentation, we can say that 2 hours of water-based ultrasonic extraction was sufficient for the extraction of more than 50% of anthocyanins from blackcurrants. We also concluded that 400 milliliters per ton of pectinex, in this case, the pectinase used, would be preferred for higher extractions of polyphenols from blackcurrants. And the positive correlations I talked about before for the extraction of condensed tannins and total polyphenols with the time of the activity of the enzymes, and also correlations with temperature used to ferment these products. So some conclusions there. And now I'm going to move to the lactic fermentation. Why do we want to ferment berries with lactic bacteria, lactic acid bacteria? Well, you know, anthocyanins could promote the growth of bacteria due to the prebiotic activity. The growth of probiotics, then the metabolism of the phenolics could give us higher antioxidant capacity. Fermenting the berries have been shown to improve sensory experience for consumers undergo by biotransformation of the phenolics and organic acids. Remember, I told you blackcurrants are astringent, sometimes bitter. So sensory can improve due to the activity of these biotransforming, processes in phenolics and organic acids. And also, in addition, fermented products with live culture can benefit human health. So it could improve antioxidant capacity and maybe other potential health benefits. The specific objectives of this part of the research were to determine and compare enzymes that are present in lactic acid bacteria. So which enzymes are present? Are those going to facilitate the extraction of certain bioactive compounds? So we compare 2 enzymes, beta-glucosidase and kinase activity of 7 commercially available bacteria. And again, the collaboration and synergy with companies such as Probi from Sweden, help us to have bacteria that have never been tested. So what we produce is unique discovery and information because nobody has done it in the past. So fermented black com was one of the berries selected, and we fermented those with lactobacillus strain from 0, 4, 6, 10, 24, and 48 hours. And then analyze different physicochemical parameters, but also chemical parameters and some biological activities before and after fermentation. I'm going to share with you some of the results. This has already been published, so except for one chapter in a book that is coming soon from the American Chemical Society, everything else is already published. So here you can see the students gathering, collecting, harvesting the black currants. Fresh black currants were used and were collected during the summer 2020 from the Department of Crop Sciences at the University of Illinois here in Urbana-Champaign. And at the end, we selected this variety, black com variety, because we found that it was intermediate with many of the parameters that were analyzed. So we decided to use this black com variety to move on with the fermentation process. Although we have also found in the past these two varieties, Consort and Coronet, as outstanding sources of bioactive compounds. So we had options. We decided to go towards this one that is intermediate in physical and chemical characteristics of the black currants, viscosity changes and different compounds present. So the results of this study can help us determine which berries, which variety is the best to use in a desirable food product. Now, this is a diagram of the experimental strategy that we use for the lactic acid bacteria fermentation. After using the berries, so in this case, we only use the juice to ferment. The juice was inoculated with different strains of, the correct name is Lactibacillus strains, and we finally use these 4 that are here and determine different chemical parameters as anthocyanins, total tannins, condensed tannins, polyphenols, antioxidant capacity, and some characterization of the chemical compounds present. I want to share this slide with you again to emphasize the importance of collaborations between academia and industry. This company, Probi, provided us with different varieties of these bacteria to ferment for the first time this black currant. But look at the properties that these bacteria have. They already know that this bacteria has a positive effect on the immune response, reduce stress related to inflammation, have anti-inflammatory properties. This other, L. plantarum Hill 19, that as you will see later was one of the best bacteria that came out of our investigation, is antihypertensive. There is science regarding these properties. And others, as you can see here, decrease inflammation or reduce bacterial translocation, some that are not very well studied yet. So there are many opportunities for discovery here. And also this other Lactobacillus paracasei, you will see it later in our results, was one of the best to produce phenolic acids and other very high antioxidant capacity compounds. They already knew that induce the immune system and anti-inflammatory properties. So very important to look into collaborations with industry again. So the fermentation method we use, first of all, adjust the pH. So we will have this Lactibacillus plantarum strain growing properly. And we analyze for the enzymes first, the tannase and beta-glucosidase activities. And then after fermentation, we analyze for phenolic composition, antioxidant capacity before and after fermentation. So you can see here the different bacteria used versus a control. Of course, we always had a control with no bacteria, and these were the times that they were fermented. And I don't know if you will see a change in color when I click here. So this is before fermentation, and these were the changes in color in the juice after fermentation. So there is one effect due to temperature, probably 37 degrees, but also the metabolism of the bacteria, the different bacteria used on the phenolic compounds present in the blackcurrant. So this is how the experiment was run. Rebecca Kowalski ran this experiment. The tannase activity and glucosidase activities were selected, and all the strains tested, all the bacteria tested contain glucosidase and tannase activities were detected in all of them. In the case of tannase activity, we didn't see a statistical difference. In the case of beta-glucosidase activity, there were differences. So what did we do? What we did was to select 4 of the bacteria to move on with the fermentation in the extremes, right? So we have very high beta-glucosidase activity and very low beta-glucosidase activity to learn if this has something to do with the results we were going to obtain. Moving forward, we will see how the pH change decrease over the time of fermentation. These are 20 hours, 40 hours, 48 hours of fermentation. And most probably you cannot detect the change, but this is 0 time, 4 hours, 6 hours, 10 hours, 24 hours, 48 hours. So maybe you can see the change in color from 0 to 48 hours of fermentation. So there were metabolic transformations taking place in this very after lactic acid bacteria fermentation. So let's start with few results and then some conclusions. The antioxidant capacity increased by L. plantarum Hill 19 and LB7 strains. So the control, as you can see here, this is the control. All of these data are changes in antioxidant capacity. The control degraded due to 37 degrees that were used, and also the change in pH. Antioxidant capacity significantly increased due to 2 bacteria. You can see here, LP Hill 19 and this other one, LB7. So what does this mean? This means that there was a significant improvement in the antioxidant capacity due to the presence of certain enzymes in these bacteria. So most probably the enzymatic activity of the bacteria cause a formation of phenolic acids and maybe other metabolites during the fermentations, which are good antioxidants. So it's important to select the bacteria that will optimize the antioxidant capacity in the juice. What about total polyphenols? Well, in this case, what we saw was an increase in total polyphenols due to the presence of L-paracasase. This had a greater ability to deglycosylate more glycosylated phenolics, removing the glucose, right, the sugars from the anthocyanins and tannins, and increase just for this variety, for this strain. So this L-paracasase had a clear effect on the increase in phenolic acids, as we learn later. And for tannins, well, the condensed tannins broke down after the addition of bacteria and were transformed into smaller compounds. Control degraded, as you can see here. This is the control, right? Probably due to pH and also due to interflavonoid bond cleavage. So the control, the condensed tannins decreased significantly. And since these tannins decrease, is good because remember I told you that the berries are astringent. So we are hoping that the sensory evaluation of these products will improve due to the decrease in condensed tannins. Maybe the astringency, maybe the bitterness could decrease in black currant juice. due to the fermentation with some of these lactobacillus, as we did in this experiment. Finally, total anthocyanins were not affected by fermentation. After 24 hours of fermentations, anthocyanins decreased, but had not a significant change from the control. So when we look at the change of total anthocyanins, we notice that there were not a statistical difference affected by fermentation for any of these bacteria. So that will give you a general overview of what we did. And let me give you in particular the summary of two bacteria here, LP HIL19 and L. paracasei that we have here. What was the effect of time of fermentation on different phenolic compounds? So what I show here is the change in the concentration of phenolic acids and quercetin from the baseline and the control, considering baseline and control. So we see L. paracasei, this first one, this block that you can see here horizontally. We can see the different phenolic compounds, phenolic acids, and quercetin, and how they change during fermentation. So let me tell you that this L. paracasei had a clear effect on the increase of all phenolic acids measured: protocatechoic acid, cumaric acid, gallic acid, ferulic, caffeic, when juices were fermented for 24 and 48 hours. The antioxidant capacity, all of them have antioxidant capacity. Of course, quercetin is one of the best, and maybe followed by protocatechoic acid here. Protocatechoic acid and quercetin could be two of the highest, the compounds with the highest antioxidant capacity. And as we can see here, lactobacillus had also been shown to metabolize phenolic acids by the carboxylases or reductase activity. And that could be the reason that there was a decrease in phenolic acids. As you can see here, there seems to be a decrease in some phenolic acids. So probably some of, for instance, caffeic acid could have been converted to dehydrocaffeic acid. And that's very bioavailable in humans, and it has a higher antioxidant capacity than caffeic acid. So in summary, we can see that these two strains of lactic acid bacteria favor the fermentation, because at the end, we were able to quantify or at least detect these phenolic acids with very high antioxidant capacity. So in conclusion of this lactic acid fermentation, we can see that blackcurrant cultivars vary in chemical composition, and they can be successfully fermented by lactobacillus strains. More research is needed, of course, to optimize the best conditions and associate those with health benefits for humans and animals as well. But we need to optimize and identify those health benefits for humans. In this case, we concluded that from the list of bacteria that we tested, L. plantarum HIL19 and L. paracasei are the most promising strains for increasing antioxidant capacity or total polyphenols after fermentation under the conditions we use in this research. The fermented juice can be used in many products. This can be added in yogurt, ice cream, smoothies. What else? You may have many more ideas of where this fermented material, very rich in antioxidants, could be added. So that concludes my presentation. But first of all, I want to talk about some opportunities and challenges and what our take-home messages here. So natural pigments are compounds in food that can be used by the food industry as additives, especially anthocyanins, right? Instead of using synthetic colorants, why not use natural pigments such as anthocyanins from berries or corn or beans or many other products that could be economic and beneficial for the consumer? There is evidence that the effectiveness of anthocyanins as antioxidants with potential effect to prevent oxidative stress in our bodies, because every day we're exposed to different toxicants in the environment that will cause oxidative stress. The development of functional foods with value-added properties is of great interest, not just to the scientific community, but also to the food industry and to the consumer. So what is the impact and future directions? Well, for the food industry, the impact is the potential to use anthocyanin-rich extracts for natural pigments as value-added ingredients and provide understanding on the effect of phenolic composition on health benefits. And future directions, well, we need further analysis on, for instance, the response of the immune system. How is the immune system going to respond to all these bioactive compounds? How processing will promote the liberation of compounds that will improve our immune system. We need additional animal studies, but also well-performed clinical studies with berries in general, blackcurrants in particular, because there are not many studies on that. But we have to be careful on characterizing the materials that we use for clinical studies. Otherwise, we will never be able to compare among different studies. And of course, the effect of processing techniques on anthocyanins and phenolic composition in plant foods. That is still needed, and there are many opportunities with so many products in Mexico. For instance, all the colored corn, all the common beans with different colors, that the different pigments, anthocyanins, tannins could be extracted and studied. So thank you again for your attention, for your time, and I'm happy to answer any questions. And again, I hope you visit us one day in Illinois. Thank you.
Thank you very much, Doctor. So we have time for some questions. Do we have? Yes.
No.
Dr. Belmont? Yes, so she can hear you.
Dr. De Mejia?
No, I cannot hear. Could you please?
Dr. De Mejia?
Yes.
Maybe you need
Can you hear?
Yeah, I think so.
Hi, Dr. De Mejia?
Yes.
Can you hear me?
Yes.
Amazing, amazing work. Congratulations. It's amazing. You cannot see it, but I have my mouth. It's open. It's just because of the mask that you cannot see it. Amazing work. Amazing work.
Oh,
I do have a couple of questions. First of all, which was the method that you used for the evaluation of the antioxidant capacity?
Okay, different methods. At the end, we end up with the ORAC method, the scavenging capacity of free radicals, but there are different methods, and you have to identify what is the mechanism of antioxidant capacity, because if you only use one method, you could conclude that your material doesn't have antioxidant capacity, but it could be that it's through different mechanisms of action. For instance, you could use free radicals, of course, scavenging of free radicals, but you could use also iron, the transformation of exactly 3+ to 2+, or you can use free fatty acid oxidation. So it's recommended to use different methods. In our case, at the end, we end up using ORAC method for scavenging capacity.
Interesting. Usually, well, I also work with antioxidant capacity of polyphenols in the form of curcuminoids. So that's why I was really interested in your talk. Have you ever considered to analyze your samples with cyclic voltammetry?
No, yet. No, not yet, but that sounds very interesting.
Actually, I think it would be interesting to try them with cyclic voltammetry for many reasons. Maybe for calculating the half potential wave, that could give you a whole of insight about the actual electrochemical potential that they have.
Fantastic.
Right? They can also currently I'm also working in predicting them with DFT calculations. So that's also another possibility that I think it would be very interesting for your compounds to be tested. They are
Right.
small molecules, or they are in the scope of being calculated with quantum mechanics with no problem.
Wow,
But what
fantastic.
I think that the biggest possibility here is to see how they react with the superoxide anion.
Mhm,
And that
mhm.
can easily be tested with cyclic voltammetry. So I don't know if you are interested in maybe collaborate with that or something.
That sounds very exciting because it will save time, effort, reagents, and maybe you can also use that method in humans, right? Can you?
Absolutely, yes.
Yeah. Mm-hmm. Oh no, that's fantastic. Absolutely. Congratulations if that's what you are using, because that sounds very exciting. Time saving and money saving, right? We don't
Exactly.
need
So,
to spend so many hours.
especially time saving in the lab.
Exactly,
Very good.
exactly. Absolutely. You have my email. Please contact me. I'll be very happy to see potential collaborations.
Oh, absolutely, Dr. De Mejia. Thank you very much.
Thank you.
Yes, Jacqueline, can you go?
My intern is going also to ask some questions, Doctor.
Yes.
Hello, doctor.
Yes, I can hear you. Thank you.
I would like to ask, for how long can your product be stored? Because I know it was freeze dried, and I don't know if that takes away some properties, or how long could you store it? Yeah, regarding the storage of the black currants.
Yeah, that's a great question. Not in this study, because we wanted, for the first time, that we were fermenting with all these bacteria, we wanted to know what were the chemical changes. So we wanted to be careful about that. That's why we freeze dry them. But in real life, most probably you will not freeze dry the materials. You can use other ways of drying the materials, but that's a really good question, because we have done studies with other berries and with other products, and you have to really protect them against light and sometimes even oxygen, because they can oxidize easily. I haven't done it yet for these specific fermented products. We keep the wine for the one that was produced, the dealcoholized wine. We still keep it at 4 degrees centigrades. Those are not freeze dried, those are in bottles, and they are in the refrigerator. For the lactic acid fermentation, we have all of them as powder after freeze drying, but we haven't done the how do you call it? The storage studies. We haven't done it yet. Thank you for your question. And I mean, if someone is interested to do it, we have the materials.
Thank you. And sorry, do you think that, for example, regarding the berries, you could harvest them and freeze dry them, or store them in some other way and use them at a later time?
Yes, we freeze them. We always freeze them.
Okay.
They are kept frozen for a long time, for a very long time, just frozen. Yes.
Okay.
Yeah.
Perfect. Thank you very much.
Oh, you're very welcome. Thank you.
Do we have any other questions here? No, well, I do. Dr. De Mejia.
Hey, Miguel.
Hello. I would like to know if you have done some studies about the stability of the products that you have. I mean, the color.
The color?
Yes, the changes in color.
Yes,
Have
yes.
you done
Yes, we have done it. And I mean, some of the slides try to show the changing color from a very strong red color, very strong red, they became pinkish, not completely degraded. I don't know if I can go back and show some, but not completely degraded, but there were pinkish color, very acceptable, still very acceptable colors at the end of the fermentation. But as I said before, we have not done a systematic study on the change in these parameters through storage, let's say after 3 months or 6 months or 1 year. We haven't done those studies, Miguel. we just stop after producing the fermented products. In the case of lactic acid bacteria fermentation, in the case of alcoholic fermentation, we keep them in bottles at 4 degrees and the color remains the same for many months now. I mean, the student graduated, almost 2 years ago, and the containers are still there with no change, obvious change in color. But there is need to study these products after lactic acid bacteria fermentation. We haven't done it. It seems like the colors after fermentation are not as strong as the original black currants, but the color is very acceptable to be added to different foods like yogurt or like ice cream or something like that.
Okay, thank you very much. Do we have any other question? If not, well, we want to thank Dr. De Mejia for her talk today. I'm very pleased to hear you again because she was my advisor during my master and my PhD. So it was a pleasure to see her again. Yeah.
Great, Miguel.
So thank you very much. I'll ask you for a clap for her.
Thank you. Thank you for the invitation, Miguel. I really appreciate, and congratulations. You are pioneers. You are pioneers in Querétaro, in Mexico, right? Bringing Arkansas University there to Mexico. So congratulations, and congratulations on all your work. I hope we can collaborate in the future. Thank you.
I hope so. Thank you, Dr. De Mejia.
Bye.
Thanks, everybody.